Good evening, ladies and gentlemen, especially if this is your first visit to the Royal Society, and a particular welcome to this evening's Francis Crick Award Lecture. I should introduce myself. I'm Jean Thomas, the Biological Secretary of the Royal Society, and I have one duty to do, which is to ask you to please switch off your mobile phones, because the lecture is being webcast and so on, and we don't want the phones going off. So please turn your phones off. So the Francis Crick Lecture is given annually in any field in the biological sciences. Preference is given to genetics, molecular biology and neurobiology, the general areas in which Francis Crick, who was a Fellow of the Society, worked, and also to fundamental theoretical work, which is the hallmark of Crick science. The lectureship was endowed by Francis Crick's friend, Sidney Brenner, also a Fellow of the Royal Society, and the first lecture was given in 2003. The recipient this year is Dr. Matthew Hurles, group leader at the Wellcome Trust Sanger Institute at Hingston, just outside Cambridge. Matt was an undergraduate in Oxford, then went to Leicester to do his PhD with Mark Joblin on the population genetics of human Y chromosome polymorphisms. And for those who don't know, the Y chromosome is the male-specific chromosome. During his subsequent postdoctoral work in Cambridge on population genetics and molecular evolution, he established the molecular mechanism underlying a recurrent deletion of part of the Y chromosome which causes male infertility. 10 years ago, Matt joined the Sanger Institute in Cambridge, where he's currently leading efforts to apply genome-wide resequencing methods to improve the diagnosis of rare diseases and to understand the factors influencing rates of germline mutation. He was chosen to be the recipient of the 2013 Francis Crick Award for his outstanding contributions to understanding structural variation in the human genome, the mechanisms that cause this variation, and its medical and evolutionary consequences. So, ladies and gentlemen, I'm very happy to present Dr. Matthew Hurles to deliver his lecture on the interesting title, Mutations, Great and Small.
Thank you. Well, thank you very much, Jean. It's a very great honour to give this talk. And it was also a very great honour to meet Francis Crick, as I did as an undergraduate. When he came to give a talk at the Oxford Union, and I was writing some very poor articles for the student newspaper at the time on science, and had the pleasure of speaking with him. So before this talk, I tried to look through pictures of Francis Crick. Most of them involve him looking at a double helical structure. That wasn't the Crick that I met. The photo that I found that most captured my memory of speaking to him was this one here, with kind of drink in hand, very convivial, being somewhat gossipy about other scientists in the room. And so it was a great pleasure to speak with him, and I was very pleased when I saw that the Oxford Student newspaper doesn't have any online archives, so I can't show you and you can't see the rubbish that I wrote about what he spoke. So the two things that I really want to talk to you about today are the work that we've been doing on gains and losses of DNA that we all have in our genomes. And then secondly, the way in which mutations arise as we pass on our genomes, our DNA, from one generation to the next, from parent to child. So the human genome can be seen at a kind of bird's eye view down a microscope. And what you see here are the 23 pairs of chromosomes that all of us have. And there's two very special chromosomes down the bottom here, the X chromosome, this rather large chromosome here, and the Y chromosome here, this rather stubby, small portion of the genome that I did my PhD on. And there's a few things that one can tell just from this bird's eye view of the human genome. The first is we can tell that it's human, because there are 46 chromosomes here. Our closest relatives, the chimps and gorillas, they have 48 chromosomes. And not just the number, but the banding pattern of the chromosomes is recognizably human. The second thing that we can tell is that this is a male, that the presence of the Y chromosome, which determines maleness. All of the men in this room have an X chromosome and a Y chromosome, and all the women have two X chromosomes. The third thing that we can tell about this individual is really from what's not there. This individual does not have Down syndrome or Edwards syndrome or Patau syndrome, the three human diseases that are caused by the largest mutations that we know about, where there's a whole extra copy of a chromosome, which we'll be able to see at this kind of bird's eye view. But those are pretty much the largest mutations that we see and all that we can really glean about this genome from the bird's eye view. And historically, we've had two views, the bird's eye view and the kind of worm's eye view. So this is what the 3 billion letters of DNA in the human genome look like close up. There's 4 of them, A's, C's, T's, and G's, and it's the order of these that's the code for life. Now, this particular snippet here is not actually a random set of A's, C's, and T's and G's. This is actually a portion of the gene FOXP2, which was described by a previous Crick lecture. And actually, one thing that's notable about this gene is even though you're only looking at a small portion of the gene, you can tell that this comes from a human as well, because there are 2 key spelling differences of single letters that humans have that are also specific to humans that other mammalian species don't have. But the question exists, what are we missing by looking at the genome from just these 2 views, the top-down bird's eye view and the bottom-up worm's eye view? So we can, by using the analogy of geography, we can think about what we might be missing. So these 2 pictures here are pictures of 2 cities in the United Kingdom. One is the city Cambridge, where I now live, where I did my postdoctoral training, and the other is the city Leicester, where I did my PhD studies. Cambridge is renowned for its architecture. It's a beautiful city. Leicester, when I first went to Leicester, I read the Let's Go Guide to Britain, and its description of Leicester's architecture was that it's unfortunately suffered the triple whammy of wartime destruction, uninspired post-war development, and industrial decline. But that's not immediately obvious from this view here. Close up, this is on the left is a snapshot of the building that I did my PhD in, and on the right, the building I did my postdoc in. Now, one could hazard a guess from these worm's eye view of geography which of these is the city with the beautiful architecture and which is not. But you may be wrong. You may not be very confident about doing that. And that's because between these 2 scales of looking at geography, we're missing out all of the architecture. How are the components put together? And with those 2 views of the genome, we're probably missing the architecture of the genome as well. So what I introduced to you in the beginning was 2 opposite ends of the scale in which a genome, all the DNA in a cell, can differ between individuals. So on the left-hand side, you can see that, hopefully in red, there's a single base change that differentiates the top sequence from the bottom sequence. And that's the smallest type of change we can have. And on the right-hand side, we've got an addition of an entire chromosome, and that's the biggest kind of change, as we see in Down syndrome. But in between, there's actually a continuous distribution of variation between these scales. So at the bottom end, there are losses of a few bases, gains of a few bases, but with the worm's eye view that I showed you before, we can see this. What we can't really see at either of the scales is the kind of variation where there are large segments of DNA that are lost in this example or gained in this example. And this is the type of architecture of DNA that we're missing when we look at it down a microscope or if we look at the individual sequence of the bases close up. So if we think about any type of variation, we need to think about what's its likely impact. Do we really care if we've lost segments or gained segments of DNA? Well, of course, our chromosomes contain a linear combination of genes, and those genes encode the proteins, which are actually the molecules in the cells that do all the work. And this flow of information unidirectionally from genes to proteins is what Francis Crick called the central dogma of molecular biology. But these genes do not occupy most of the genome. Actually, there are about 22,000 genes that we're aware of in the human genome that code for proteins, and they only occupy between 1% and 2% of the genome. 98 to 99% of the genome does not code for genes, and we are still in the very earliest stages of creeping out of our ignorance of what these sequences that are not genes, what they do. What we do know is not all genes are turned on in every cell of the body, and that's why the cells are different. Each cell expresses a different set of genes, and the sequences that lie between the genes are clearly involved in the regulation of those genes deciding which genes to turn off when in what cells. So if we think a little bit about how this form of variation that we haven't really been able to ascertain previously, these large segments that are gained and lost, if we think about what kind of effect they might have on the genes, well, it's reasonably obvious that if we took a cartoon of a chromosome on the left with 3 genes in here, then we might have variants that removed an entire copy of a gene, the loss of gene B in this example. Or we might have variants that gave us an additional copy of gene B, as in this example. There are other forms of variation which I'm not going to be talking about, which don't change the numbers of genes but change the orientation of genes. And I'm going to be focusing on the gains and losses of DNA that collectively are called copy number variation. Now, the mechanisms that generate this copy number variation are actually blind to where the genes are in the genome. So I've shown you examples of removal or gains of copies of a gene, but equally they could be removals and gains of other portions of the genome. And it's not simply that one can lose or gain existing functions in the genome, but one could potentially generate new genes. For example, here at the bottom is a deletion which is making a new gene, and it's the hybrid of gene B and gene C. So there's the potential not just to toggle on and off existing forms of function, but actually generate new forms of function. And we can see in the evolutionary history of our genome that many of the genes in our genome have undergone this kind of process. So in thinking about this talk, I was very much reminded of a quote from Sidney Brenner, who you heard about earlier. And Sidney, ever since I learned this quote, it's really stuck with me. Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And the key thing is that last segment, because science is often portrayed as a very hypothesis-testing kind of approach. You think of an idea and you go out and you do the experiment. But that's to underplay the value of exploring the new vistas on nature that new technologies give you. And I think the examples I'm going to tell you about today very much exemplify what Sidney was talking about. Because we were very fortunate that new techniques came along about 10 years ago which enabled us to probe the genome to look for these segments of DNA that might be gained or lost. And this technology involved this picture on the left is what's known as a microarray. So it's a glass slide, a microscope glass slide, which has small portions of our DNA spotted on it. And the color of those spots is telling us whether there is more or less of that segment of DNA in a given individual. And most of them are yellow, which tells you that most of us, for most of our genome, have exactly the same amount of DNA, but there are a few spots in green or in red which are regions which have been gained or lost. And by applying these technologies and applying them to looking at normal individuals, normal populations, we could make new discoveries. And this picture is a map of the gains and losses of DNA that we found in normal individuals. So you'll hopefully recognize the chromosomes here, and every blue line tagged onto those chromosomes is a gain or loss of a DNA in the first map that we generated of these losses. And this was in collaboration with my long-time collaborator, Nigel Carter, who's since retired. And he really drove this new technology, and together we made these new discoveries. So the important thing about this is it suddenly gave us an insight into the fact that actually everyone in this room does not have the same genome. We don't even have the same number of genes. We probably vary by several hundred genes between us, each of us not having some and having others. And this is much more extensive than we previously thought. Some of these variants are very common in the population, and some are very rare, but there are hundreds of genes that are affected. So when we discovered this, I was somewhat disturbed to see the headline in the newspaper, "The Book of Life is Rewritten." Slightly disturbed about this because this suggested that we'd done some kind of genome engineering. The Book of Life had done nothing at all. It had just sat there, same as it ever was. What had happened was that we'd just reread it, and our understanding of it had got better. So it was striking that there were all these differences, that we are somewhat more different from each other than we thought. But the question remains, should we really care about them? Are they biologically important? Now, one can investigate this biological importance at different scales of biology. One can look at cells, one can look at individuals, and one can look at populations. So by looking at cells, I don't have time to go into all of it, but one can essentially say, I know this cell does not have a copy of this gene, or only has one copy of the gene rather than two copies, because we should have two copies of every gene, one inherited from either parent. And one can go in and look and say, well, do we see less of that protein that gene encodes in that cell? And typically we do, but not always. And if we have extra copies of that gene, do we see more copies of that protein? And typically we do, but not all the time. So there's clearly a molecular change that results in the cell from that change in the DNA. But we still don't know whether the cell or even the organism cares about that molecular change. There are molecular differences between all of us. So what we really care about as individuals is, are these going to affect our health? And so I'll talk a little bit later about investigating whether this type of variation plays an important role in disease. But first, I'll talk a little bit about populations. So if a type of variation is important, then we should see it under natural selection, under Darwinian selection, winnowing out harmful mutations and potentially increasing the frequency of beneficial mutations. So what happens if we look at CNVs in this way? So we can think about purifying selection, which is the selection that's winnowing out the harmful mutations. And we can think about positive selection that's amplifying the beneficial mutations. And in this context, it's interesting to think about those copy number variants, deletions and duplications that affect genes on the right, and those that fall outside of genes on the left. Do we see a difference between the two? Because we know genes are important. If we don't see a difference in the distribution of these variants between the genes and the non-genic regions, then we're probably thinking that biology doesn't really care about this form of variation. But when we look at the effect of this purifying selection, we can see that it's acting very strongly. Because remember, the mechanisms that generate this variation are blind to where the genes are. So these variants are being generated all the time. But the variants that fall within genes, they're getting removed from the population by natural selection. And we see there are many fewer, although there are many hundreds of variants that affect genes, there are many fewer than we'd expect. And the ones that we do see are typically less common in the population because they are being kept down by natural selection. If we look on the other side, positive selection, well, most variants in the human genome, if they change function, change it in a negative sense. And that's because we have a highly evolved DNA. It's pretty good at doing its job. Evolution over billions of years have managed to achieve that. So most changes that you make are not going to be beneficial. So purifying selection dominates. But we do find examples for both copy number variants that affect genes and copy number variants that fall outside of genes, which show clear signatures of being selected positively by natural selection. Now, we can see those signatures, but we don't necessarily know why that is. There's one really nice example which our collaborator Charles Lee worked up for a particular type of genic copy number variant. So the gene that encodes the protein that digests starch in our saliva is called salivary amylase. And that gene exists in multiple copies. And what Charles showed was that populations that have prehistorically been eating high-starch diets have more copies of that gene. And populations that historically have had low-starch diets have fewer copies of that gene. And presumably, there has been positive selection for those additional copies to drive the digestion of starch in saliva. So moving on then to diseases. Well, we can broadly break down diseases where genetics plays a fundamental role into common diseases such as diabetes or coronary artery disease, and we can break or rare diseases such as cystic fibrosis. And I'll take common diseases in turn first. So common diseases, we know genetics plays a role, but it plays a role in concert with the environment. So the genome and the hamburger. And what we know about the genetics of common disease is that the genetic variants that influence our risk of those common disease are spread throughout the genome for any one disease that we might think about. There are many tens, probably hundreds of variants that influence our risk of getting type 2 diabetes, for example. But each one of those variants only has a very modest effect on our risk, maybe increasing it by 10 or 20%. It's certainly not causing diabetes. And those modest effects, those subtle effects, we can only see those effects if we look at very large numbers of individuals, and so we can only really detect these effects if these variants are common in the population. So there's a fairly standard way of trying to identify if a variant affects the risk of a common disease, and that's just to take a set of individuals with the disease and a set of individuals without the disease and just look at the frequency of that variant between those two individuals. And so one can simply, in this case, label the individuals who have this variant in both the patients and the controls. And in this situation, as in most times we do this kind of experiment, we see there's absolutely no difference. This particular variant doesn't influence the risk of diabetes. So as a large-scale UK-wide consortium, we investigated whether thousands of copy number variants from the maps that we made in normal populations, whether they influenced the risk of 8 different common diseases. And we found very little. We identified 4 copy number variants that influenced one of those 8 different diseases. And all 4 of those were already known previously. And so we were left with a rather surprising conclusion that despite the fact that these variants are very large and they remove large segments of DNA, very, very few of them actually influence our common disease risk. But it's a different story when we come to the genetics of rare diseases. So the genetics of rare diseases, there's no hamburger on this picture, because the genetics of rare diseases is driven by very strong mutations that in and of themselves are sufficient to cause those disorders. And broadly, they can be classified into 2 types. Those where just a mutation in a single copy of the gene is sufficient to cause the disease, and those where, like cystic fibrosis, both copies of the gene need to be damaged before the disease occurs. So in collaboration, so there are many different rare diseases. There are thousands of rare diseases. There has been estimated, though, that although each one is individually rare, about 1 in 15, 1 in 20 of us has a rare disorder. So it's cumulatively quite a big impact. We focused just on one study in collaboration with Sadaf Farooqi in Cambridge, who has been for a long period of time been working on the genetics of extreme early onset obesity in children. So you'd say obesity is a common trait, but the extremeness and the early onset of what the patients that she works with is actually a much rarer trait. And we applied exactly the same kinds of technologies that I showed you to generate those maps to the patients that she works with. And we found that there was one particular region of a chromosome, of chromosome 16 in this case, where we found 2 different types of deletion, losses of DNA in these families. We found a small one where about 200,000 of these letters of DNA were lost, and we found a big one where about 1.7 million of these letters were lost. And if we looked at the families of those individuals, then that small deletion that we observed, we observed in children in families where there were multiple individuals with extreme obesity. They often had siblings, and they had at least one parent who was morbidly obese. Whereas the very large event that we observed that overlapped that, we only found in individuals where they were the only person in their family to be morbidly obese. And so we think that there are particular genes within this interval that these 2 deletions share in common that are very important for the sensing of when we're hungry and when we've had enough. And so we looked at these CNVs and we tracked how they passed down through the families. And what we observed was, and that's shown with these green lines here, is that in the families where there were multiple individuals who were morbidly obese, every individual carried this deletion that was morbidly obese. But in the families where the individual was the only one affected, these were new mutations that the child had that neither parent had. And that explained why these individuals were the only members of the family that were obese. Now, we can't necessarily treat these particular diseases at the moment, but a diagnosis is very powerful, these families. And in this particular example, it was extremely powerful because the families at the top, several members of those families, several children in those families have been taken into social care because they've been regarded as a case of parental neglect. You've got a morbidly obese parent, often morbidly obese sibling, and you've got another child becoming morbidly obese. But after these results were fed back to those families, those kids were then given back to the families. So can't be treated, but obviously had a major impact on those families' lives. So this is just one example of one rare disorder where we found that these copy number variants were important. But the types of copy number variants we're talking about here are not common. They're very rare. In fact, they're extremely rare. Fewer than 1 in 1,000 individuals will have this, one of these rearrangements. So, and that's what we see in common with many different rare disorders, is CNVs are not necessarily the only cause, but they account for an appreciable portion of many of the rare diseases. So why do we see this difference? Why are rare gains and losses of DNA important in rare disorders, but pretty unimportant in common diseases? So I think the reason is probably just down to the sheer weight of numbers. So if we think about common diseases, so remember these are the genetics of common disease, we can only look at the common variants, those that have a subtle effect on the risk of disease. And if we look at all the common variants in the people in this room, we'll see that the vast majority of them are not CNVs. For every common CNV, we've got 1,000 other common variants of other types. And all of those have been through the filter of natural selection. They've only become common in the population because they're not highly harmful to us. And so it's perhaps unsurprising that when we look for common variants that influence common disease, CNVs do not play a major role. But if we look in rare disorders, if we look at rare variants that essentially knock out an entire gene, then actually copy number variants probably account for 1 in 10 of those. And that underscores why probably 10% of rare disorders or rare diseases are caused by copy number variants. So we see this interesting dichotomy. CNVs are clearly biologically important, but it's really the rare ones that are playing the major role in human disease. So I want to move on to the second part of my talk now, which is about mutation rates. So I alluded initially in that obesity study to how new mutations can be important for disease, but new mutations occur in all of us. All of us have mutations in our DNA that our parents did not pass on to us. And we need to think a little bit about the journey that DNA takes as it goes from one generation to the next. So this, you may not recognize it, is you at a very early stage in your life when you were just a single cell after the fertilized egg. And we know that as you pass on your genes to the next generation, you pass it on through your sperm or through your eggs. But how does the DNA get from that original single cell down to the sperm and the eggs? So what happens is you get early development, you get division of these cells, copying of the DNA each time the cell divides, because each cell has the same DNA component as each other, until we reach the primordial germ cell. And here, the paths of men and women split. So to create eggs, there are an additional set of genome copyings, but essentially all the eggs that a woman has in her life are in her ovaries when she is born. And all that happens during the menstrual cycle is that one of those just matures and so that means that every single egg that a woman produces, the DNA in that egg has been copied the same number of times, roughly about 30 times. But it's a different story with men. Men produce sperm throughout the course of their life post-puberty. And so what you see here is at puberty, a particular special form of cells called spermatogonial stem cells form. And these turn over every 16 days in the testis. And so 23 times each year, the DNA is getting copied. And that means that the sperm of a 40-year-old man has DNA that's been copied probably twice as much, maybe even 3 times as much as the sperm of a 20-year-old man. So if we think that this copying process, like all copying processes, is prone to error, and maybe that's where mutations come from, then it suggests not only that most new mutations might come from dads rather than mums, but also that the number of new mutations might increase as a dad gets older. And that hypothesis was made a long time ago because you could make that hypothesis as soon as you understood this journey of cells from one generation to the next. But we can use the tools that we have to measure mutation rates and see if this is really correct. So there's a number of different strategies one could take to measure mutation rates. In concept, they're all very simple. All you're really doing is comparing the DNA of one generation to previous generations. So on the left-hand side, you have comparing the genomes in the sperm of a man to the rest of his DNA. In the middle, you have the obvious example of sequencing the DNA of a child and comparing to their parents. But equally, one could look at the mutations that have accumulated over evolutionary time by taking two species that have had a common ancestor. And that's a relatively straightforward experiment to do. And it highlighted when the chimpanzee genome was sequenced and compared to the human genome, some very intriguing observations. So focusing on this one evolutionary approach first, what was observed was looking specifically at these single letter changes and the differences between humans and chimpanzees. And what was observed was that the number of differences you see between humans and chimpanzees depends on which chromosome you're looking at. So at the top here, you've got the X chromosome. In the middle, you've got all the other chromosomes bar the Y chromosome down the bottom. And if you compare the humans and chimpanzee genomes, what you see is on the X chromosome, there's a difference between humans and chimps about 1 every 100 letters of DNA. But at the bottom, the Y chromosome, there's a difference about 1 every 50 bases. So this suggests the Y chromosome has been mutating twice as fast as the X chromosome. And the other chromosomes are in between. Now, if we think a little bit about how these chromosomes are passed on from one generation to the next, this fits with the hypothesis that I mentioned before that most mutations come through the male line, because mothers pass their X chromosome on to their daughters and to their sons. Fathers only pass their X chromosomes on to their daughters. And that means the X chromosome's journey through evolution spends 2/3 of its time in the female germline and 1/3 in the male germline. And if we compare that to other chromosomes, the non-sex chromosomes here, they spend an equal amount of time in the male and female germline, but the Y chromosome is only ever passed on from father to son, and it has the greatest divergence between humans and chimpanzees. So this very much fits with the idea of males being more mutagenic than females. So through this kind of study and others, we have the picture of what the average genome might look like. So your genome here, were we to sequence it now, we would find there would be about 3.5 million variants that differentiate your genome from the person next to you, and indeed from the reference human genome. If we focus just on the genes, we'd find a much smaller number. We'd see about 10,000 variants in those genes that would affect the protein that those genes produce, so a much smaller number. And if we focused specifically on how many of those 3.5 million variants are new mutations, we would find that each one of us has somewhere in the region of 50 to 100 new mutations in our genome that our parents didn't have. Now, most of these mutations are these single spelling errors, single base changes. And what we know is that from the studies comparing humans and chimpanzees and others, we know that the mutation rate for a given letter of DNA is about 1 in 100 million generations. Now, that's pretty good, given all the number of times that DNA has been copied going from one generation to the next, and only 1 in 100 million bases is being mutated. But 100 million is a big number, but it's a lot smaller than the number of humans on the planet today, 7 billion. And what that means is actually every single base in the human genome, in the reference sequence, is mutated tens of times in the humans that are currently living on the planet today. Now, if we think about other forms of variation, so the gains and losses that I mentioned to you before, then actually these occur at a much lower rate. So it's probably only 1 in 20 of us in this room will have a deletion or a duplication of a segment of DNA that's longer than maybe, say, 1,000 bases that's new that our parents didn't have. And we can understand a bit more about that process by taking this different strategy for measuring mutation rates that I mentioned before, comparing the sperm of a man to the rest of his DNA. And what we need to do this, because each mutation process is vanishingly rare, the types of deletions that I'm going to describe to you mutate a bit more rapidly than a single base. It's not 1 in 100 million, it's more like 1 in 100,000. But we have to design assays that are essentially capable of picking out the one sperm that has a deletion against a background of 99,999 sperm that have no deletion whatsoever. And a really fantastic postdoc who worked in the lab, Dan Turner, actually designed 8 of these different assays to look at different portions of the genome. And I'm going to describe to you what he found. So what he did is he looked at 4 regions of the genome where we know that there are deletions or duplications that cause genetic disorders. And he designed assays for each one of these rearrangements to work out how fast these are occurring in a man's sperm. And what he found was that the most rapid one was occurring about 1 in 24,000 sperm, this deletion here. The slowest one, this duplication here, was about 1 in 1 million sperm. Now, the individual sperm donors that Dan looked at were just normal men drawn from the population. They didn't have a genetic disorder. That means that all of the men in this room have sperm in their testes now, swimming around with these deletions and duplications in them. But we could also discern something of medical relevance from this, because we could compare the frequency with which we see these mutations in sperm with the frequency of the disorders that they cause. And if we looked at 2 disorders where we think they're pretty well diagnosed, these disorders, and we have a good sense of how frequent they occur in the population, then they actually agree very well with the mutation rates that we identified from sperm. But if we looked at 2 other disorders, here shown in green, actually what we observed was the mutation rate we observed in sperm is considerably higher than the frequency with which these disorders are being diagnosed. And so we hypothesized that actually these are being missed, these are being underdiagnosed, and subsequent studies have shown that to be the case. And we also investigated this very rare event here, which had never previously been reported in humans, but we thought probably, from our understanding of the mutation process, would exist. And because this duplication is very similar to other duplications that we know cause disorders, we predicted it would cause a particular type of developmental disorder. And subsequent to us publishing this, Jim Lupsky's group have gone on to show that's the case. And what this shows is that if you have a good understanding of the biology of mutation, you can predict diseases that you haven't observed yet. So we also wanted to think about mutation as a biological process itself, a bit like height. We know that height is influenced by environment. We know that height is influenced by genetics. Is it that mutation rate is also influenced by environment and genetics? So we looked at multiple different sperm donors for one particular rearrangement, this fairly rapid deletion that I mentioned before. And we saw quite a lot of variation. But these are all quite rare events. You have to sift through millions of sperm to find a few tens of events. So is this variation just random or not? So we thought one way to answer this is by looking at sperm from twins. So if we look at identical twins, and they have very similar mutation rates, that suggests there's something systematic that's influencing those mutation rates. But if they have dissimilar mutation rates, that suggests that genes and possibly the shared environment is not so important. So we measured the rate in the first twin and the second twin of an identical pair. And if there's no relationship between these two, we'd expect to see just a flat line. There's no obvious relationship. If the first twin has a high mutation rate, the second twin could have a low mutation rate. But what we actually observed was the opposite. If one twin had a high mutation rate, the other twin had a high mutation rate. Now, it's quite hard getting sperm from twins, harder than you'd think. Quite often because one of them's had a vasectomy. And it's impossible to get sperm from twins that are all the same age. So we've got pairs of twins that are different ages. So if age is an important determinant of mutation rate, then maybe that's just explaining why some of these twins have very similar mutation rates. So we looked at that, but actually what we observed was there's absolutely no relationship between the age of a sperm donor and the rate of deletions that occur. So this is a different mutation process from the one I described previously of single base changes, and this one doesn't appear to have a relationship with age. But if we want to understand the mutation process that's most prevalent, those that's generating the single base mutations that I mentioned before, then we need to do the very obvious experiment of looking at children and comparing to their parents. And so we've done this, and it's a quite simple experiment. What you essentially do is you have mother, father, and child. You have a DNA sequencing machine, hopefully one with a nice neon light on, and you just look for the new mutations that the child has that the mother or father doesn't have, and then you need to use a few genetic tricks to try and work out which ones came from dad and which ones came from mum. And the first two children that we looked at in this way as part of a large-scale international consortium called the 1,000 Genomes Project, these kind of cartoons represent an awful lot of work, but essentially in the first child that we looked at, we could indeed see most of the mutations, and each one of these dashed lines indicates a mutation. Most came from the dad, and a small number came from mum. And then we looked at the second child, and we actually found that more of them came from mum and fewer from dad. And so there's quite a lot of variation between both the mothers and how many mutations new mutations arose on the chromosomes that they passed on, and between the fathers. So we thought we need to try and explore this variation. Why is there this much variation? Well, some of it could occur just purely by chance, because we're looking at relatively low numbers of events, 50. So if the average number of mutations in my sperm at this moment in time was 50, not every single one of those sperm would have 50, some would have 40, some would have 60. And if you kind of think about the mutation process and how it works, broadly speaking, it's quite reasonable to expect quite a lot of variation between the sperm, even from one man at one point in time. So what we wanted to do was to try and get around this variation, was to look at families where the same mum and dad had had kids over quite a period of time. This is all unpublished data. So we looked at 3 families that had had 4 children over more than a decade's worth of time. And the simple idea was we want to compare the numbers of mutations that each child got from mum and dad and see whether it changes between the oldest child and the youngest child. And what we observed in these 3 families, so each one of these lines is the number of mutations we saw in the children in each one of those 3 families as the father got older. So you can see in each one of these families, there is a clear relationship between the age of the father and the number of new single base changes that we see in the children. But strikingly, it's not the same pattern between the families. So these 2 families here, every year that the father gets older, his sperm appear to be acquiring 3 mutations per year. Whereas this family here, it's fewer than 1.5 mutations per year. And other people have estimated this in large numbers of samples, and they've estimated a population average at about 2 new mutations every year is about what we expect to see. But these evidence suggests that it may not actually be the same between families or between individuals. So we need to do more work to understand this, to understand whether there are genetic or environmental factors that are influencing these mutations. So we also have been able to look at what fraction were from the mum and what fraction from the dad, and as we expected, most are from the dad. So that hypothesis that more mutations would be from dad and that there'll be more mutations as dad got older really seems to hold true for these single base changes here, but it doesn't hold true for these, the particular mechanism of gains and losses of DNA that I showed you before. So why do we see this different effect of the father's age between these 2 mutation processes? Well, to understand that, we have to go back to the journey that DNA takes as it moves through a generation. So if we look at this process here, these small changes of a single letter, then these can actually occur at any stage in this, because these can occur any time that DNA is copied as it goes from one cellular generation to the next. Whereas what I didn't tell you about the mechanism that generates this type of variation is that it only occurs in a very specific cell division that only occurs once during the maturation of an egg and once during the maturation of a sperm. And so this type of variation doesn't have the same paternal age effect, and it also doesn't necessarily come more from dad. So there are actually different mutation processes, each one with their own properties that we need to understand, rather than a one-size-fits-all. And so just in the last few moments, I want to talk a little bit about the disease impact of these new mutations. I gave you a couple of examples earlier of those families that had children with extreme early onset obesity. But new mutations are increasingly being recognized as a cause of rare disorders, especially rare developmental disorders. And I and my colleagues, some of them here, Caroline Wright and Helen Firth, have been working on a project called the Deciphering Developmental Disorders Project, which is a collaboration with the entirety of the NHS and the genetic services within Ireland to try and see if we can use the kind of technologies that we have access to, those arrays that I showed you, the sequencing machines that we have access to that the NHS doesn't, and try and use them to diagnose children that the NHS can't currently diagnose. So the nature of this kind of clinical problem is that most of the time a child comes in to a clinical genetics centre with a severe developmental disorder, the parents are perfectly healthy. And so we can ask the question, are new mutations one of the reasons why we see most of the time the child is the only one affected? And if we understood what is the genetic architecture of these different disorders, because there are a whole set of different disorders involved here, then we could better inform the NHS what kind of technologies they ought to implement to cost-effectively diagnose these children. Now, recognising that many of these children, there won't be cures available for these disorders, but they can have a massive impact on families in terms of informing them about what the risk is of having a second child with the same disease, and potentially offering them pre-implantation genetic diagnosis to avoid having that disease in a second child. But also many of these children are misdiagnosed and they're on inappropriate treatments. And so the families are really desperate for diagnoses. And we're working very hard to try and use these new technologies to provide them. So the ultimate aim of this project is to recruit into the study 12,000 families, each with a child with a rare developmental disorder. And thus far, having analysed the first 1,000 of those families, we can diagnose about 20% of these children, just with our current understanding of what kinds of mutations can cause disease. And we're finding that most of those diagnoses are new mutations, as we might predict. And the reason we can pick those up is because in this study we're looking at the DNA of the children alongside the DNA of their parents. So most of these mutations are new, and most of them are the small spelling kind of errors of single base changes that I mentioned before. So the question then arises, we've had these new technologies, we've made new discoveries, what new ideas stem from this? Well, and this is somewhat provocative, we need to think, we can think now with our current understanding, what would it take to minimize the morbidity caused by these new mutations? What kind of things could we do as a society, or ought we to consider doing as a society that might enable us to minimize this? Well, the first thing that we understand is many of these disorders are caused by new single base changes, and we know that older men have more single base changes than younger men. So actually, one simple thing we could do would be to all donate sperm to ourselves, aged 18 or 20, freeze it, and only then use those sperm to conceive children. And of course, the problem with this is we can't just target this at individuals at risk, because all of us have new mutations, and all of our children have new mutations. So it's not possible to identify who's going to be at risk. It has to be a population-wide strategy. Because the number that I mentioned before, 100 million, 1 in 100 million generations, that's a large number, smaller than the number of people on the planet. It's also smaller than the number of sperm in the testis of every man in the room. And what that means is that in the sperm of every man in the room are sperm carrying every single disease mutation that we know about. And that means it's purely a matter of chance whether we go on to have children with developmental disorders caused by these new mutations. So it's not possible to identify those that are at higher risk, other than, of course, parental age. But of course, one doesn't have a time machine to go back and get sperm from one's 18-year-old once one's decided to have children age 40. So the other approach that one could potentially take is using prenatal screening. Now, we already screen prenatally for developmental disorders, and we do this using ultrasound. And at 20 weeks, there is an anomaly scan that is offered to parents. Most parents take this up, but this will only pick up developmental disorders that manifest themselves as some kind of large-scale structural change within the fetus that can be picked up by ultrasound. It won't pick up whether, for example, that individual might have intractable seizures for the rest of their lives or never be able to speak. It won't capture those functional deficits. And it's also very hard to counsel parents about what to do in that situation, because when there's a structural problem, it could be that structural problem comes along with all kinds of other intellectual disabilities, or it could be that structural problem is just in isolation and it could be easily repaired during the first few years of the child's life, who would then go on to have a happy life. And currently, parents who have these scans that reveal that there's a structural problem with the fetus have a very difficult decision to make, and potentially we can make that decision more accurate or give them more precise information if we added genetic screening into this. But this clearly is something not everyone will be comfortable to do, and it's not the job of scientists to tell society what society should do, but it is the responsibility of scientists to say what society could do based on our current understanding of what is causing these sometimes devastating disorders. And so these are relatively new ideas that need discussion and debate, and that kind of squares the circle of what Sidney Brenner was talking about, was that new technologies begat new discoveries, begat new ideas. And recognizing that there are many things that we do today that are historically or prehistorically would have been regarded as being absolutely abhorrent, and we take completely for granted. And this gives me a shameless opportunity to show my favourite Raymond Briggs cartoon, which is taken from Ugh, Boy Genius of the Stone Age, as he proposes to his father that perhaps floppy trousers rather than these chiselled stone pants that his father's wearing might be a more appropriate way of dressing. So I think Sidney was very much right. There is a lot of value in scientific progress to exploring what new technologies enable you to see about the world that you didn't previously appreciate, and then deriving from those new ideas that then begat further progress. And with that, I'd like to thank all of you for listening. I'd especially like to thank many of the people I didn't have time to thank in the talk that we've worked with over the course of the years. All of what I've described to you is very much a teamwork between different people, between clinicians, between researchers, between computer scientists, and it's only that I'm standing here in front of you that the hard work of all of those people. I mentioned a few of them as I went along, but there are many others, some in this room, to whom I apologize in advance. And it's also, I think, extremely important that we thank the families who contribute to these studies, both the families who are perfectly healthy and volunteer for the research that we do on understanding mutation rates, and the families who are desperate for a diagnosis and volunteer to be part of studies like the DDD studies that I mentioned before. And so last month, we tried to give back to these families in some small way through charity bike rides. We organised 9 around the country with the clinicians and the researchers involved in the project. And between us, we cycled about 4,000 miles that weekend, which is pretty much the equivalent of Land's End to John O'Groats, back to Land's End, back to John O'Groats, and back again. So these 2 family support groups, SWAN UK, that's Syndromes Without a Name, and Unique, do a completely invaluable job of working with these families to help them negotiate this tricky path of trying to find a diagnosis for their children. And the prize money for this lecture that Jean will hopefully give me in a moment will be going to this fund. And I recognise that not all of you can read that, but I have a whole set of leaflets that I'll be putting outside that contain that URL, and I'd like to encourage you to support these charities because they're very worthwhile that they're doing for these families. And so with that, I'd just like to say thank you to you again, and be happy to take any questions or comments.
thank him properly later, but Matt said he'd take questions. So if you have a question, put your hand up, and we can take several to stack them up. There are 2 microphones, one each side. So if you put your hand up, we'll direct the nearest microphone to you. And if we could have the next hand up, so that we can direct the second microphone. So here, yes?
Thank you very much for a wonderful talk. Chromosome abnormalities, you didn't really talk about them. And as far as Down syndrome is concerned, I believe it's the mother's age that's more important. I just wonder if you might comment on that kind of abnormality.
Yes, so that's very much well recognised that Down syndrome and the other trisomies do increase with mother's age, and they don't increase in this linear kind of way with father's age, as I showed you, but they occur in a kind of S shape, and so it ramps up dramatically after 35. But that's extremely well known, and it's interesting to reflect on the fact that the way in which we orchestrate prenatal screening in this country takes account of mother's age. It recognises that epidemiological relationship. There's no similar equivalent of taking account of father's age, for example. And it's an open question as to whether father's age and new mutations of single base changes is actually more detrimental than maternal age and causing those chromosomal abnormalities. But that's something that hopefully we'll be able to answer in the next couple of years.
Are there any environmental factors that can affect the rate of mutation?
So that's a very good question. So people have looked very hard and found precisely there are no recognised environmental factors that increase germline mutation rates as they pass on. But it's quite difficult work to do because the mutation rates are quite rare. There are several tens of factors that are known in mouse studies to increase mutation rate. And so we'd assume, but they're the kinds of experiments that one can't do on humans, certainly not ethically anyway. And so it's highly likely that there are environmental factors, but we can't do those experiments, so we just have to rely on natural experiments of often of our quote-unquote ingenuity, things like the Chernobyl accident or nuclear test sites or exposures to other types of environmental mutagens accidentally. Those are because the nature of those experiments is often very difficult to do them because we don't really know what dose people received. Because it's not a controlled experiment. So there are likely to be environmental mutagens, and we don't know what they are.
Any more questions? Yes? Over here, thanks.
When you're doing the prenatal screening, presumably you will only suggest rejection of a fetus or a embryo which has a recognized lesion which is going to cause a disease. You won't just ask for an embryo that's got a lot of changes which don't go in the particular areas that you recognize to be thrown away. Because if you do that, aren't you reducing, in the long term, the genetic variability of the human population and its ability to evolve?
I mean, firstly, I guess I would say that the way in which, and I think this is the right way, in which prenatal screening is done now is it's non-directive. Parents get to choose. It's up to the parents to choose what the results, and scientists, doctors, no one should really be telling them what to do. And that's all we can be doing is giving them the most accurate information to make those choices. I think the point that you made, I think, given the sensitivity of prenatal diagnosis, if it were to be implemented, it would have to be absolutely rock-solid evidence that we knew that that variant was really going to cause a completely deleterious change. But that's my view. And we already know from prenatal screening that there will be fetuses with heart defects that parents will decide to keep, and fetuses with heart defects that parents will decide to terminate. And that's their choice. And I think that non-directive view of this is very important. And I guess as to your second question about evolution, I'm not too worried about that. I think there's plenty of new mutations being produced all the time. Every single base is being mutated in the genome as we speak. So that there's plenty of fertile material for evolution.
Is there one? Yes.
I have a simple question about sequencing. When you say something is something like CCT, can it also be reset as GGA because that's the other side?
Yes, so typically we're only showing one of the two strands of DNA.
So which one do you show first? When you give them out and publish them, which comes first?
So it depends exactly on the nature of the publication. So it depends if your gene goes that way along the chromosome or that way along the chromosome. But typically, if you don't have an imposed direction, we tend to go from left to right.
Well, which of the two halves do you take, A or T?
We take the one that, so you can imagine one DNA strand going in that direction and the other going in that direction. We tend to take that top strand and report it.
The top one.
Okay, I think we're past our finishing time, so we should stop there. But I want to thank Matt for a lecture that was packed full of information, bang up to date, and thought-provoking, and I think that's reflected in the sort of reflective attitude of the audience now and the sort of questions that we've had. So I think it's been a splendid lecture, and in order to recognise that, he is going to get a cheque. But first of all, he's going to get a nice certificate. So, thank you very much. Very nice medal, the Francis Crick medal.
On behalf of the families, thank you.
And as he said, he's going to donate it to this charity, which I think is the first time we've seen anything like that. I think is wonderful. Thanks.